Okay.
So we have experiment on ultrasonication. So this ultrasonication is used for cell destruction. So as part of this experiment, we are going to find the rate constant for cell destruction. As we discussed in the theory class, cell destruction using ultrasonication is assumed to follow first order kinetics. So by doing the cell destruction for repeated cycles, okay, for a constant time, so and then plotting a graph between ln of 1 minus r versus the number of cycles in x-axis, so we can find the rate constant from the slope of this graph. So first, so this is the ultrasonicator setup. Okay. This is the control panel. So where we have the display, where we can fix the on time. So say 9 seconds, off time 1 seconds. So we have kept the minimal time, lowest time, to avoid the denaturation of your bioproducts due to the increase in temperature. Because so one of the major disadvantages in the ultrasonication for this cell destruction is when your cells are exposed to the ultrasonic waves, due to this high frequency waves, temperature of your cell suspension will increase. So it is important to provide the cooling system, and decrease the time of exposure. Therefore, we can avoid the denaturation of the product. Okay. So here we are going to disturb the same sample again and again for different time. Okay. So here time is going to be the variable. So disturb your cell slurry, say for example 30 seconds. Okay. After 30 seconds, you take 0.5 ml or 1 ml disturb the cell slurry. Okay. Centrifuge the cell slurry and find the concentration of protein present in your sample, okay, by using Bradford assay. Okay. For which you have to take 20 microliter of sample. This is a filtrate sample. Okay. Mix this 20 microliter of sample with the 2 ml of Bradford and incubate at 37 degrees Celsius. Okay. And then take OD at 595 nanometer. So you will get the OD for your samples. So after first cycle of 30 seconds cell destruction, you have to collect the sample for protein estimation. Okay. And then disturb the same disturbed cell slurry again for the second cycle. In the second cycle also, you take another 30 seconds. Okay. So total time is 60 seconds. So the cycle time is 30 seconds. Okay. So after each 30 seconds, you have to take this sample. So first cycle time is 0 to 30. After 30 seconds, you have to take sample. And second cycle is another 30 seconds. That is total time is 60 minutes, 60 seconds. And then third cycle is another 30 seconds. So total is 90 seconds. Okay. Similarly, you repeat it for 10 times. So you will have the 10 number of cycles. So the same cell slurry disturbed after each cycle is taken as a feed for the next cycle. So in this display, we have fixed on time 9 seconds, off time 1 second. So it will run for 9 seconds and then it will off for 1 second. Therefore, the temperature can be removed by a cooling system. Okay. And we have the next one is time, total time. Here the cycle time is 30 seconds. So we have to set the time. So working time is 0.5 minutes. Second. Okay. Minute nothing. 30 minutes like that. We have to fix 30 seconds. So we can fix 0.5 minute. So we have fixed the 0.5 minute for cycle time. Okay. So that means 30 seconds. And temperature is we have fixed 27 degrees Celsius. So this is the chamber where the ultrasonic wave will be generated. So this is the ultrasonic probe connected with the regulator. Okay. And this is the ultrasonic probe. So for different samples, we have the different size of ultrasonic probes. So this is kept in a small chamber to avoid the noise pollution. Okay, so therefore, if it is closed, then the noise will not come out, so we can avoid noise pollution. So this is the chamber to control the noise pollution. So inside you are going to keep a sample. So this is your sample, say E. coli culture, and it is kept in a beaker which contains ice. Okay. So therefore, when the heat is generated during the ultrasonication, that heat can be disseminated using the ice. So in this tray, so this is a movable tray. Okay, first keep this tray at the bottom. So this is the platform. And then adjust this level of platform. Okay, so therefore your ultrasonic probe will uh, will be immersed on your sample. Okay, but you ensure that the probe is not touching the bottom of the your sample container or side wall of the sample container. Okay, so now you can see your ultrasonic probe is immersed in the sample. So it is adjusted so that it is not touching the bottom or side wall of your beaker. Therefore, this will not break your beaker. Okay, so now it is closed. Then now if you press the on button, then the ultrasonication will start. So the probe will release the ultrasonic waves to the sample. So the ultrasonic wave frequency is about 20 kilohertz. Okay. So in and around say 22 kilohertz, so we have fixed it. So to have the ultrasonic waves above 200, here we have fixed the input power supply 60%. So it will be around 140 to 150 watts. So 140 to 150 watts is given as the input power to the your ultrasonic probe. Now your 30-second of cell disruption is over. Okay, so it is stopped. Okay, your first cycle is over, and then take out your sample. So lower the platform. So take out your sample carefully, okay, and then collect micropipette. So you have to use micropipette to take your sample. So collect 0.5 ml to 1 ml of your disturbed cells. So I have collected approximately 1 ml of sample after first cycles. This is supposed to be centrifuged before finding the protein concentration. So centrifuge the sample. After centrifugation, take 20 microliter of this filtrate supernatant. Mix it with the 2 ml of Bradford reagent, incubate at 37 degrees Celsius for 10 minutes, and then take OD for protein at 595 nanometer. Then using standard, we can convert the OD into the concentration of protein. Okay. So then we should go for second cycle. Okay. Cycle time is 30 seconds. So first we have completed first cycle. So now we are going for so you have to use the micropipette to collect the sample. So 0.5 ml is enough, otherwise you can go up to maximum 1 ml. So now sample is collected for first cycle, and then again keep your sample inside the chamber just above this platform, and then adjust the platform height. So therefore this ultrasonic probe will get immersed on your sample, but ensure that your ultrasonic probe should not touch the bottom of the beaker or side wall of the beaker. Okay. When it is touching the side wall or bottom of the beaker, then due to these ultrasonic waves, okay, so your beaker will get vibrated and it may break. Okay. So then we have kept the sample for second cycle. Now close it, and then switch on the setup for second cycle. Now it will go for another 30 seconds. Okay. So total time is 60 seconds. Okay, now second cycle is over. Then open the door and down this platform. Then take out your sample, or you simply can keep it inside by using your micro pipette. You can collect the sample. So now we are taking sample for second cycle. So after second cycle, so we have collected sample. So using micro pipette, you can collect the sample and take it in a F&F tube. You mark it with serial number 1, 2, 3, 4 for cycle after first cycle, after second cycle. Okay. Similarly, we should continue this process, okay, for 10 different cycles. So for third cycle we have to go for another 30 seconds. Therefore, total time will be 90 seconds. Okay. So after taking the sample, after second cycle, I kept the beaker again inside, and the probe is also immersed. Okay. Then we can go for the third cycle. Similarly, repeat this for 10 times. Therefore, you will have the 10 number of cycles. After each cycle, you will have the samples. Okay. So centrifuge the samples, okay, and then using Bradford assay, find the protein concentration in your sample. And in your first order equation, we have ln of 1 minus C by C max is equal to minus Kn. Okay. So whatever the concentration of protein obtained after first cycle, second cycle, third cycle are known as C1, C2, C3, C4, so up to C10. Okay. And then we should know the C max, that is the maximum concentration of protein present in your sample when the 100% cell disruptions happened. Okay. That C max can be determined by taking 2 ml of undisturbed cell, okay, and mix it with 2 ml of 0.19 sodium hydroxide. So boil this mixture for 10 minutes. And then take 0.5 ml of this sample, so centrifuge it, and then find the protein concentration present in your sample using Bradford assay. Okay. So using standard, you can convert this concentration into mg per ml. Okay. That is C max, which is obtained after cell disruption using sodium hydroxide and then followed by boiling. So that is C max. Then we have the formula ln of 1 minus C by C max equal to minus Kn. And the C is C1, C2, C3, C4 up to C10 and C max. Okay. Then you can find the ln of 1 minus C by C max with the number of cycles. Then plot a graph between ln of 1 minus C by C max versus n. Here the number of cycles or time in x-axis, and the ln of 1 minus C by C max. So C by C max is simplified into capital R in your manual. Okay. So by plotting this graph and finding this from this graph, you can find the rate constant for the cell disruption using ultrasonication.